This video is going to cover prokaryotic cells and what are called the cell shapes and arrangements. Really more of a lab thing as far as the cell shape part. But what happens is bacteria, when they divide, and we're talking about bacteria now, they're very simple. Like it states up here, they do not have membrane-bound organelles, and they divide by a process called binary fission. Basically, one cell splits into two cells, and it depends on what species. Some can divide every 20 minutes, some an hour, some an hour and a half. Fungus, a little slower. But what happens with bacteria, there are three basic shapes. You have circle-shaped bacteria, they're referred to as cocci. You have rod-shaped bacteria that are called bacilli. And you have S-shaped bacteria, the cells. All of these are cell shapes. They're called spirilla or spirillum. A microbiologist, the first thing you would do if you want to look at a microbe is you look at what is called what's the shape of the cells and what's the arrangement if you're going to try to identify something. On our PowerPoint, see here are the cocci, here are the bacilli. Now these are, those look more like spirochetes. Spirilla are a little more S-shaped. And what ends up happening, depending on the species, is once a cell divides, sometimes they stay together in pairs and groups and that kind of thing. More strength in numbers. Cocci, a diplococci, pair of circle-shaped cells. There's one right there. I'm going to go there. Gonorrhea, Neisseria gonorrhea. If you do a gram stain on Neisseria gonorrhea, you get a gram-negative diplococcus. The circles, the cells are found in little pairs like that. Streptococci is a chain of circles. Sarcinae is a group of 8. Tetra would be a group of 4. And then Staphylococci. Now, Streptococci and Staphylococci, yes, those are genus names for bacteria. Streptococcus causes throat infections, but this is talking about arrangements. This is not the name. There's actually an arrangement called a streptococci. And yes, you would find streptococci, the ones that make a person sick. Streptococcus pyogenes, which is a bad one. If you gram stain them, they would be in a chain of circles like that. I'm not going to worry about this Coccobacillus. It's supposed to be between circle and rod. Here's a bacillus. Here's a diplobacilli palisading when they divide, like a snapping division, and they're uneven like that. And here's a streptobacilli. So basically, you would look at the cell shape arrangements of any microbe if you're studying them. Spirilla are S-shaped. Vibrios are comma-shaped. But let's quickly go through all the parts of a prokaryotic cell. Great picture. I'm just going to stay right here. Everything is written verbatim in your outline, but here is a prokaryotic cell. This little area here is called a nucleoid. It contains one long circular chromosome. It's not a nucleus, it's a region, nucleoid region. Ribosomes are little bitty dots, and there is cytoplasm within a prokaryotic cell, just like with a eukaryotic cell. That says a food granule. Prokaryotic cells have a thing called an inclusion. They're like vesicles. They can store food, water, and air in those. Here is a plasmid, which is a circular piece of double-stranded DNA. These cover luxury genes. We're going to talk about these more later. But bacteria can actually exchange these. If you have a pathogenic bacteria that has a plasmid for antibiotic resistance, meaning the cell can produce enzymes that would break up penicillin, they can pass those on to other cells. And then you'd have a whole bunch of cells that are resistant to antibiotics. Flagella, tail, moves the entire cell. Now here's one of the big differences between human cells and bacteria cells. Human cells, the plasma membrane, that is us. It's also called a cell membrane. Eukaryotic cells have that as well. Cell walls. Now, in our example for eukaryotic, we didn't cover these. Plant cells are eukaryotic cells that have a cell wall. But bacteria have a cell wall. So it's a rigid outer layer. And this particular example, these are called fimbriae. These little things coming off, they're actually like Velcro. They help a cell stick to something. But a capsule or glycocalyx or slime layer, the sticker layer, not all bacteria have that as well. This prevents phagocytosis. In other words, they can't be broken down by white blood cells. It also allows them to stick to a surface. That's why you have to brush your teeth, is because you have capsule-forming microbes in the mouth, and they form a film, and then they stick to teeth. So there are I was talking about the inclusions. And I'm going to go through the last part of this, and there is a thing called an endospore. That's Joseph Tyndall came up with a way of killing these things, and as well as staining them. Spores are dormant structures, and this example, I'm sorry the writing is so small, but basically there are some species of microbe, if they're about to die, they can put their DNA and essential cell parts into a spore, which is right there, and then the rest of the cell breaks off. So you have what's called a vegetative part of the cell that's active. Spores are dormant. And that spore can sit there for a long time. They've had documented cases of spores sitting for thousands of years until the conditions improve and they can break out of that and start growing again. Cell membrane, we did cover that with the eukaryotic cell. It's the same thing as a eukaryotic cell membrane. It's 2 layers of phospholipids. Like I said, cell wall is a rigid outer layer. Now, this is one thing that is different. Bacteria have, and I'm going to go through our lesson here, bacteria actually have 2 different kinds of cell walls. And it has to do with when you stain them and how penicillin affects them. Let me go to the next page. Yeah, here we are. So, as far as organelles go, they have ribosomes. They're very simple. Here's an endospore example, and then here's a cell membrane or plasma membrane. Wow, looks like that's no longer active. But so here's an example of, and we'll get to this later, the cell wall. There are cells that have what's called a thick layer of this substance called peptidoglycan. And they're called gram-positive cell walls. That's a staining technique. And you have to do the staining technique to figure out if you have a gram-positive. Some bacteria have one that have a cell wall that's made of a lipopolysaccharide and then a small layer of peptidoglycan. And I was hoping I could find, there we go. I'll leave it on this. There's a procedure called a gram stain. We cover this in lab, where you do a staining technique and you figure out, and it puts it into 2 categories, gram-positive or gram-negative. This looks like an example of a gram-positive cell wall. See the thick layers here. Here's the cell membrane underneath. And then here's a gram-negative cell wall. This is called the lipopolysaccharide layer, and here's the stuff called peptidoglycan. And it just, it puts, basically you have microbes that have this kind of cell wall, and you have one that have this kind of cell wall. Here's the thing, penicillin, the drug, it actually binds to peptidoglycan and prevents it from forming. So that's how it kills bacteria. So gram-positives that have this kind of cell wall, they're killed pretty easily by penicillin. Gram-negatives, not so much. I mean, they can be, but that's when you have some modified medicine to help get through, which is like essentially an extra cell membrane. Capsules, I briefly talked about that. Appendages, yes, there are flagella, bacteria move. And depending on, and this is not human beings, only sperm have a tail as far as human beings go. There are different arrangements for the flagella. You have microbes that have several on one end. That's the Lophotrichus arrangement here. Monotrichus is one. Peritrichus, well, one on one end of the cell. Peri could be all the way around. Like here's a peritrichus. Here, this is a Lophotrichus, and that's a monotrichus. And then there's atrichus, which they have none. But flagella allow them to move. The ones obviously like this, they can move a lot faster than the ones that have less flagella. So I hope this helped. I hope the guided tour of the prokaryotic cell helped, and thanks for joining me.
And that's the bottom line.